Right now, deep in your midbrain, clusters of dopaminergic neurons are firing, releasing dopamine into circuits that control motivation, reinforcement, and goal-directed behavior. But how does this system actually function at the molecular level, and what happens when addiction can hijack it? The dopaminergic system originates in the ventral tegmental area, or VTA, where dopamine neurons project to several brain regions, forming key pathways. The mesolimbic pathway, which connects the VTA to the nucleus accumbens, is central to reinforcement and reward processing. Dopamine release into the nucleus accumbens enhances synaptic plasticity through D1-like receptors, which activate G-protein-coupled cascades, leading to pKa-mediated phosphorylation of AMPA receptors, increasing synaptic strength. In contrast, D2-like receptors couple to GI proteins, inhibiting adenylyl cyclase and reducing excitability, fine-tuning dopamine's effects on behavior. Under normal conditions, dopamine is tightly regulated by the dopamine transporter and by presynaptic D2 autoreceptors. Specifically, dopamine transporter rapidly clears dopamine from the synaptic cleft, while presynaptic D2 autoreceptors inhibit excessive firing of VTA neurons via GIO-mediated suppression of voltage-gated calcium channels. This maintains a balanced reward response, where natural reinforcers like food and social interactions generate moderate, transient dopamine surges. But drugs of abuse overwhelm this finely tuned system, producing supraphysiological dopamine spikes that disrupt synaptic homeostasis. Things like cocaine and amphetamines block dopamine transporter, preventing dopamine reuptake and prolonging its presence in the synapse. Opioids like heroin and fentanyl act indirectly by binding to mu-opioid receptors on GABAergic interneurons in the VTA, inhibiting tonic inhibition and disinhibiting dopamine neurons, leading to excessive dopamine release. This chronic overstimulation induces profound neuroadaptive changes. D1R-mediated signaling in the nucleus accumbens enhances expression of immediate early genes like CFOS and FOSB, transcription factors that drive synaptic remodeling. FOSB accumulation in particular promotes dendritic spine growth, increasing the strength of drug-associated synapses while weakening natural reward circuits. Simultaneously, prolonged dopamine exposure leads to internalization of D2 receptors, reducing inhibitory control and increasing compulsory drug-seeking behavior. Addiction also alters glutamatergic transmission from the prefrontal cortex to the nucleus accumbens. Chronic drug use reduces presynaptic glutamate release while enhancing postsynaptic AMPA to NMDA receptor ratios, shifting synaptic strength toward a hyperexcitable state. This dysregulation in corticostriatal circuits contributes to impaired executive control, making relapse more likely. At the cellular level, addiction is essentially a pathological form of synaptic plasticity, characterized by long-term potentiation of drug-associated circuits and long-term depression of natural reward circuits. However, targeted interventions can reverse these changes. Exercise, enriched environments, and pharmacological approaches targeting glutamatergic homeostasis, like for example NMDA receptor modulators like ketamine or mGlu-R2/3 agonists, have shown promise in restoring synaptic balance and reducing drug cravings. So ultimately, addiction is a consequence of neuroplasticity gone awry, hijacking the same molecular mechanisms that allow us to learn and adapt. So whether reinforcing a habit, chasing a goal, or battling compulsive behavior, dopamine circuits define what we pursue. And at the molecular level, every choice we make is, in some way, a reflection of how our neurons encode reward.